Let me share my screen. Put this in play mode. So thank you so much, Jay. It's really a pleasure to be here and to participate in this phenomenal seminar series. Thank you so much for hosting it and initiating this great effort during the past several months. So as some of you may know, our laboratory is interested in understanding the structure and function of large ATP-dependent chromatin remodeling complexes. Most notably, the mammalian switch SNF, also called BAF, family of chromatin remodeling complexes. And we place particular emphasis on understanding how perturbation to the individual components of these complexes can play driving or contributing roles in human disease. So our laboratory is largely centered in biochemistry, biophysics, increasingly structural biology, and we integrate in epigenomic and genome-wide genomic approaches, as well as systems biology and even machine learning, to really take a full constellation of approaches to identify the various ways by which this molecular machine can be perturbed in disease. And in parallel, I'm fortunate to partner with and participate in groups intensely dedicated toward translating such discoveries into hopefully meaningful therapeutics for patients, which brings me to my disclosure slide. I'm the scientific founder, board of directors member, SAB member, shareholder, and consultant for Foghorn Therapeutics, which is a company I started a few years back. And I will not discuss any off-label use or investigational use in this presentation. So I think this group really needs no introduction, but of course the sequencing of the human genome and subsequently cancer genomes has immeasurably advanced our understanding of the molecular underpinnings of human disease. And just a couple of years ago, we actually saw the fruit of a very large scale effort called the Cancer Genome Atlas, TCGA, where in the key paper shown here on the bottom, these authors reported the sequencing of over 33 different cancer types and a number of course, subtypes within those, over 10,000 tumor samples, and they identified 300 driver genes and over 3,000 putative missense mutations. It's really quite a remarkable effort and continues to serve as the inspiration for much of what our lab does. And I'm sure this is true for a number of the other groups watching this talk, that really human genetics in the context of human cancer have really inspired a wide range of new studies in the area. This is actually one of my favorite panels from the large series of papers published in Cell Press, where you see here on the x-axis a large number of cancer types, all abbreviated by their TCGA nomenclature. And you can see on the y-axis a number of signaling pathways or processes that are perturbed, with the color bar representing the frequency of genes in those pathways mutated in any given cancer. So we see mutations, of course, in signaling pathways. These are signaling pathways with components that have been long-standing culprits for various drug discovery efforts, both by pharma and academia for many years. You see mutations in apoptotic and cell cycle regulators. But perhaps the most surprising and unexpected result to come from all of the sequencing studies was the previously unappreciated idea that chromatin regulatory processes, epigenetic processes, are frequently perturbed. In fact, some of the most frequently perturbed across human cancer. So you see here by the bars I've highlighted, there are mutations in histone modifiers, proteins that can create histone modifications, DNA modifiers. The complex I'm going to tell you most about today, the switch SNF complex, is so frequently mutated, it gets its own row. It's called its own pathway among the various ones shown here. And of course, we also see mutations in transcription factors, which can leverage various facets of the chromatin landscape to elicit their cell, tissue, and ultimately disease-specific phenotypes. So as I mentioned, you see mutations in genes encoding proteins and protein complexes that render chromatin in this closed conformation largely inaccessible to transcriptional machinery, and thus the genes controlled by these regions of chromatin are held in the off position. And we see mutations that are in proteins that enable the accessibility of DNA, allowing DNA to become open and open for active transcription. So you see these mutations such as DNMTs, TET enzymes, for example, DNA modifiers, histone modifiers, histones themselves, so actually mutations in the very octamer itself, histone tails, and for the purpose of this talk, mutations in chromatin remodeling complexes such as the switch SNF complex. Now, this is just an artistic representation of the mammalian switch SNF complex shown here, drawn approximately to scale with its local nucleosomal environment. These complexes utilize the energy of ATP hydrolysis as its engine to essentially mobilize DNA nucleosome contacts, thereby enabling timely and appropriate DNA accessibility throughout our genomes. These complexes were originally discovered in yeast, in screens for mate-type switching and sucrose fermentation. They were later characterized in Drosophila, and then ultimately in the mammalian cell context, and actually even most recently by our group in defining the various complexes that are contained within this family, and finding that there are actually 3 distinct final forms, then working to order how these complexes are pieced together, their assembly modules, etc. So on the left, I show you the 29 genes that encode the various members of these complexes. But you can appreciate from the drawing that there aren't 29 subunits, and that's because these subunits are actually pieced together in a combinatorial fashion from the products of those 29 genes. So this gives rise to a large number of combinatorial possibilities, even in one given cell. We have multiple types of BAF complexes running around the genome, all comprised of slightly different subunit componentry. So you see that because of this combinatorial assembly, there are many hundred total possibilities that the way that these things can be assembled. There are 3 final form complexes, again shown here for simplicity, just one of them, with specific subunits that can demarcate each final form. And there are mutually exclusive paralog subunits. For example, 2 different ATPases called SMARCA4 and SMARCA2, 2 different ARID components called ARID1A, ARID1B, and so on. So the reason that we are so excited about these complexes is indeed their frequent mutations in cancer, as I mentioned on the previous slide. And if we now take a deep dive and look at TCGA, even the most modern updated TCGA, you can see that if we sum the mutation frequencies for each of the subunits across human cancer, this is PanCan, you can see that the mutations are present in over 20%, around 20% of human cancers. And if we start to now add in addition to exon mutations, you start to add in loss of heterozygosity, deep deletions, you can see that this number continues to rise. So here, over 24% of human cancers, just the mutations in the exome. And of course, there are this is still the tip of the iceberg, because exon mutations are just one way to perturb a gene. There are, of course, other mutations throughout the enhancer regions, other intronic regions that can also control the expression of these genes. So this is really a major burden across human cancer. So one of the reasons I actually am familiar and tied to Alex's Lemonade Stand Foundation, and one of the reasons I find my home department at Dana-Farber and Harvard Medical School as pediatric oncology, is because in a number of rare but genomically well-characterized cancer types, for example, those shown here on the x-axis, in a number of those, the mutation in a component of the mammalian switch-NF complex is the hallmark uniform feature, the driving feature, and as you can see, near 100% of cases in many of these types of tumors, for example, synovial sarcoma, which I'm going to tell you about today, has this characteristic SS18SSX fusion protein, which is found universally in essentially 100% of tumors, synovial sarcoma tumors. There are a number of other genes that encode for components within the chromatin remodeling complex I'm describing, which are, again, exhibit uniform or near uniform driving features in these cancer types. So you can see some of them are recurrent. You can see SMARCB1 present in malignant rhabdoid tumors, atypical teratoid rhabdoid tumors, which are also pediatric conditions, epithelioid sarcoma, other pediatric chordomas and epithelioid schwannomas also have pretty high frequencies of mutations in this gene. So with this, these are a number of rare cancers that really provided strong support for an initiating role, the ability of this complex to play a driving role in the initiation and maintenance of these cancers. And it's many of these that we've essentially used as platforms for us to better understand the overall structure and function of these complexes. So what I've told you is that specific subunits can be mutated in specific cancers, as you see from this bar chart and in the heat map in the previous slide. Other subunits can be mutated across a much more wide range of cancers. For example, ARID1A is the most frequently mutated subunit within this family of complexes. And of course, if that's mutated in a wide range of cancers and a wide range of tissue types, this suggests some defining commonalities. Intriguingly, some of the most frequently mutated subunits are actually not those that are required for the textbook definition, the canonical definition of these remodelers, which is to remodel chromatin. Essentially, a lot of the assays that we use in this field leverage the ability to take a complex and place it on a chromatinized template and just allow it to do its job to essentially remodel those DNA nucleosome contacts. But it turns out that not all of the subunits are actually required for that function, begging us and the rest of the field to really identify new assays that can more faithfully recapitulate mammalian chromatin architecture so that we can really understand what those components are doing. And then finally, some of these mutations can be heterozygous. So this really implicates the importance of gene dosage effects throughout cancer and also other diseases. So over the past 5, 6 years or so that I've had my young lab, we've essentially walked our way around this complex, if you will, essentially focusing mostly on rare pediatric tumors, such as many of the ones shown here, where again, you can see the very frequent percentages of tumors that have a specific perturbation to this complex. So we've done some work on loss of function settings, for example, examining the deletion of certain subunits. And this is a characteristic one right here, the SMARCB1 deletion, which is deleted in essentially 100% of malignant rhabdoid tumors and atypical teratoid rhabdoid tumors, and over 90% of epithelioid sarcomas. Another type of rare ovarian cancer, small cell carcinoma of the ovary, hypercalcemic type, which has as its defining feature dual loss of both of the ATPases, so the entire engine of the complex is missing. We've worked to identify the biochemical modules and structural modules, in fact, a 3D structure generated using cryo-EM, cross-linking mass spectrometry, was just published last week in Cell. We've worked to define the functional modules, as well as the interactions of these complexes with transcription factors. And those transcription factors can be those that are overexpressed in disease, or even other fusion proteins, which are characteristic of other pediatric malignancies, such as EWS-FLI1 in Ewing sarcoma. And these factors tether transiently to the surfaces of these regulators to drag them essentially, or hijack them to new sites on the genome. So across all of these settings, one of the very unique disease contexts that we've studied, and actually it was my initial foray into chromatin remodeling, was this synovial sarcoma condition. And this is, again, 100% of synovial sarcoma cases have this oncogenic chromosomal translocation, which results in this fusion oncoprotein. And by virtue of this fusion oncoprotein, SSX is added on to the SS18 subunit shown here. And we've done a number of work, and I'm going to tell you a new story that emerges from our work on this in the lab. So this actually dates back to some of the work I did as a graduate student while I was in Jerry Crabtree's lab as a cancer biology graduate student several years ago, where we actually identified SS18 as a novel component of the complex, and then tried to examine what happens in tumor cells that now have this fusion oncoprotein, SS18-SSX, which again is driven from this TX18 chromosomal translocation that is pathognomonic for synovial sarcoma. What we found was that this fusion protein usurps these BAF complexes by dominantly integrating into the complex, displacing the residual remaining SS18 from the remaining allele, wild type allele, and somehow, and it was unknown at the time, seemingly displacing this subunit called BAF47 or SMARCB1, the same tumor suppressor subunit that's deleted in malignant rhabdoid tumor from the complex. And the net result of all of this is that we found at the time, back in 2013, that this complex would then target to sites that had some of this H3K27 trimethylation, and would activate genes. In this case, it was still in the era of doing qPCR, so just one that we focused on, SOX2, and that this would support proliferation. Excitingly, we found that we could actually toggle between this oncogenic form of the complex and its wild type counterpart by simply just changing the concentration of either the fusion protein or the wild type SS18. And that would dictate whether the complex had the fusion protein in it, the wild type protein in it, and it would change its behavior on chromatin, and it would also cause for the cells to either continue proliferating or quiesce. So what we wanted to do is to understand then what happens if we perturb the levels of the oncogenic fusion. And here you can see on the right-hand side of the slide, just using a shRNA mediated knockdown approach, you can see a very clean deletion of the two bands corresponding to the oncogenic fusion. And this is paired with a concomitant increase in SS18 wild type levels, as well as the BAF47 tumor suppressor subunit, SMARCB1, shown here, increasing levels when we get rid of this fusion. So this begs the question, how would this oncogenic fusion affect the global targeting all throughout the genome? How does this affect the global targeting of BAF complexes and subsequent gene expression programs that are elicited by its targeting? So to do this, again, we use this shRNA approach, and you can see here in synovial sarcoma cell lines, in the presence of the knockdown, you can see that complexes measured here by 3 distinct antibodies that bind to and tag the complex. You can see that these complexes disappear from a very seemingly cancer-specific set of sites that is only present upon the fusion incorporation. So with the fusion being present in these synovial sarcoma cell lines, you can see this top part of these sites are really very specific for the synovial sarcoma setting. And when we release that, when we remove the fusion, you can see the complexes are released to target to a whole set of other sites on the genome. And this was really quite intriguing because it was the only disease setting at the time that we had found where there was such a stark difference between where complexes go on chromatin when the fusion is there and where complexes go when the fusion is not there. In many of the other cases, you lose sites in these loss of function tumors. So this was really very unique in what we were observing here. This could really be moved around. You're just changing the distribution of complexes on chromatin. And you can see that in the reverse experiment on the right as well. If you just take fibroblasts and we now introduce either the wild type protein or this SS18-SSX protein, we can see here that the complexes marked by SS18 are again dragged to these new set of sites. And this is also paired with increased accessibility at those sites. So we're really changing the chromatin landscape by simply adding this fusion. Another interesting thing that we noticed is that these complexes, when we examine where do they go on the genome, that's what I showed you on the past slide, we noted that they were going specifically to regions of chromatin that are supposed to be repressed. They're supposed to be repressed by complexes such as PRC2, polycomb repressive complex 2, that keeps these genomic loci in the off position and heavily repressed. So you can see here both measuring the complex occupancy. So here we're looking on the top panel of each of these, you see wild type SS18, and on the bottom panel, you see this fusion oncoprotein, which forms these very clear, very dense localizations. These are actually at inactive X chromosomes in these cells. These are bar bodies, so they're very known to be very heavily repressed. And you can see the occupancy co-overlap, or the localization co-overlap with these members of PRC1 and PRC2 complexes, respectively. And you can see that also on the genome-wide level. What we noticed, which was again unique for this cancer setting, was that BAF complexes overlap with PRC2 complexes across the genome. So much so that if we now look at on the left-hand side, the Venn diagram here in synovial sarcoma cell lines, if you just try to map where is this repressive complex shown here marked by SUSY12, a component of PRC2, it is overlapping much more strongly with BAF complexes in the control condition. So when we have not knocked down the fusion, but when we knock down the fusion, you can see many of those sites go away. And vice versa in fibroblasts, when we introduce the fusion, you can see much more overlap at these SUSY12 marked sites. So this brings us to a key question, asking what would be the mechanism then that would make these complexes go to these very site-specific regions of chromatin? Why is it going to these repressed sites? And if it is going to these repressed sites, what is the molecular basis underlying the interaction of this complex with chromatin? What is the actual mechanism by which this can interact with chromatin? We like this question because if we understood this and we could answer this, we could certainly define the specific features of the interaction of the fusion protein with chromatin, but we could also understand cancer-specific synthetic lethal dependencies that have been observed in synovial sarcoma through these larger scale genome scale screening studies. And most importantly, we could inform and design new therapeutic approaches based on these basic mechanistic findings. So to start, we started with a biochemical approach, as we often do in our lab, and we simply purified complexes containing SS18-SSX and complexes containing wild type SS18. So they're just purifying these BAF complexes using these tag subunits shown here. And we noted something very striking right off the bat when we did that. You can see that when we purify wild type complexes, they come off chromatin very easily. There's very little in the chromatin fraction, which is abbreviated here with CHR, and they're nice and soluble in the nuclear extract. You can pry them off chromatin, no problem. However, when we try to do this same experiment with SS18-SSX, you can see that it was nearly impossible with these experimental conditions to pull off these complexes from chromatin. And they were so tightly held to chromatin that you are actually observing, you're purifying out the histone octamer. These are members of the nucleosome shown here at almost 1-to-1 stoichiometry. You're actually capturing the complex and the nucleosomes to which it's bound, suggesting that they're bound very tightly, and very little comes out in the nuclear extract. If we now run these on mass spec experiments to try to quantitate some of those peptides, again, you see that in the context of the SS18-SSX fusion, this is really enriching for members of the BAF complex, as well as many components that are encoding for the various histones. So this was quite striking. We then tried to use additional biochemical approaches to see if we were recapitulating this no matter which way we tried it. And indeed here, using 10 to 30% glycerol gradients, these are just simple measurements of size, of density sedimentation. So heavier proteins will migrate toward the right-hand side of the screen, toward the heavier fractions. Lighter proteins migrate toward the earlier fractions. On the top part, I'm showing you a very normal profile of BAF complexes. So you can see they're migrating roughly around fractions 13 and 14. You can nicely label all the components in the complex. But look what is the stark contrast between that and the SS18-SSX1 shown on the bottom. The gradient looks like a mess. The complexes are purified are actually higher in molecular weight. These are the ones that are coming out as full complexes. And they're pulling with it the members of the histone octamer. You can see here members of H3, H4, H2, H to be coming out with these complexes. So suggesting it's very hard to pry once the complexes have this SS18, SSX, it's very challenging to pry them away from their nucleosome substrates. Taking another approach yet, differential salt extraction, one can use to try to identify how tightly held are these complexes on chromatin. And again, you can see that they're very challenging to remove from the chromatin pellet, even at 1,000 millimolar salt. But the second we start to remove the fusion protein from these synovial sarcoma cells, you can see that they're released into the more soluble fractions, and they can be precipitated at lower millimolar salt. And finally, you can use imaging studies based on FRAP to identify the residency times of these complexes on chromatin. So you can see that the synovial sarcoma associated complexes have much higher residency times. They're sitting on chromatin longer, they're more tightly bound, and they're dissociating at a lower frequency. So that was all in examining now the entire complex as a whole, with or without the fusion protein. So to try to isolate what was going on, we determined that the best strategy to do that would be to remove SSX from the fusion protein itself. So really, since this is the main thing that's added, this SS18, this SSX tail onto SS18, let's study that tail in isolation. So just take the 78 amino acids. So we did that and purified the 78 amino acids, shown here just tagged to a GST protein for solubility. And what you can see here is that when we purify this and now subject them to binding studies with nucleosomes, you can see that the 78 amino acids just by itself is sufficient to pull down histones. It's sufficient to actually capture histones, suggesting a binding interaction between SSX itself and chromatin. So this was surprising and previously unknown. We didn't know that SSX would be able itself to, tether to and engage the histone octamer. So then, to try to get a better idea of this, we subjected this SS18, this GST SSX protein to nucleosomes. This is now mammalian nucleosomes taken from, for example, 293 T cells. So they have all the constellation of marks on them, and we're just simply incubating those with the nucleosomes and performing some quantitative targeted mass spec. And what we found here was that indeed, though the SSX protein, if incubated with nucleosomes, selectively purifies and captures out nucleosomes that have these repressive marks. So we were capturing nucleosomes much more frequently that had these repressive marks than those that had activating marks. And you can see here with an activating mark like acetylation, as you add more and more and more acetylation groups, you see less and less and less and less binding of the SSX 78 amino acid tail to the nucleosomes. And this is relative to H4 unmodified, where you have nice enrichment of those nucleosomes. So to try to examine what's going on with the 78 amino acids in a bit more detail, we simply performed some alignment studies, taking the 78 amino acids and aligning throughout the SSX family, as well as a related family known as PRDM7 and PRDM9. And what we noted when we did that is just in this last 34 or so amino acids of the 78 amino acids, there was a basic region and an acidic region. Basic region shown here with a lot of these blue colored amino acids, and this acidic region with here a lot of these negatively charged amino acids. We found that the 34 amino acids are sufficient to bind these histones, and then proceeded to make mutations throughout this 34 amino acid region to identify where in this 34 amino acid region, what are the regions most required to actually bind the nucleosomes, to bind histones. And what we found here is that if we made any perturbation in this basic region, so for example, this lysine here, 168 to an alanine, or simply swapping out all of the alanines for basic amino acids, you could see a complete disappearance in the ability to even bind histones. So it seemed like that basic region is very important for even binding the nucleosome itself. However, if we made any mutations in the acidic region, or even swap that entire acidic region for alanines, you could see that the binding to histones was still maintained. So this region we didn't yet have an explanation for, but this basic region we knew was critical for binding the nucleosomes. So then we zoomed out a little bit to try to understand what happens, what's the result of all of this in cells. So we turned to ChIP-seq approaches to map these complexes on chromatin. You can see here the SS18 SSX protein. Again, I'm showing you that very unique set of sites to which it targets. And this we found by doing these studies that the 34 amino acids itself was also sufficient to target it to these very specific sites on chromatin. However, any mutation that we made, 24 amino acids, so not the full complete 34, a deletion of the acidic region, a deletion of the basic region, all of these were insufficient to target the complexes to these specific sites. And actually, as you can see from the heat maps here, they looked very similar to the wild type SS18. If we now translate that ChIP-seq and try to examine what happens at the RNA-seq level, you can again see that SS18 SSX or SS18 SSX with the 34 amino acids was sufficient to elicit this expression pattern where some genes here are very strongly activated and others are negatively regulated. And you can see that again, any of the perturbations to either the acidic or the basic region were unable to elicit that effect. And finally, this carried over into proliferation. We know that these synovial sarcoma cells are dependent on the presence of the fusion protein. So we did perform some rescue experiments to examine which types of the fusion were able to rescue these cells in terms of proliferation. You can see that only the full-length fusion with the 78 amino acids or this 34 amino acids were able to rescue. And this is compared then to any of the versions of the SSX that didn't have the full 34 amino acids. So they were missing either the acidic or the basic regions. So this basic region I'm showing you seems to disrupt nucleosome binding. So what happens on nucleosome targeting? Well, we pursued to make a single residue mutation, here shown the R169A mutation in that basic region. And again, this is responsible for tethering to, this region is important for tethering to nucleosomes. So if we make this mutation, you break the interaction. And then if we examine what happens on chromatin, just the single point mutation alone is able to break the synovial sarcoma specific signature as to where these complexes go. To get a little bit more of a handle on where on the nucleosome this is actually binding, we teamed up with Tom Muir and his talented postdoc, Hai Dao at Princeton, who had developed a very clever technique using photocross-linking. So you actually take reactive diazurine probes that are strategically placed throughout the nucleosome histone octamer, the H2A, particularly the H2A, H2B interface. So it's in a way to examine the acidic patch. And then incubate your protein of choice. In our case, we incubated SSX. And what we found here was that there was striking photo cross-linking to H2A-E56, and that could be ablated by removing that arginine and turning it into an alanine. And similarly on H2B, the E113 residue, that could also be significantly attenuated by either mutating the tryptophan or the arginine in that area of the basic region. And in reciprocal studies, we took and purified the 78 amino acids of SSX and then incubated them with either nucleosomes that have wild type composition or nucleosomes that have a problem in the acidic patch itself to see whether or not SSX would be affected in its binding. And indeed, if we take any of these nucleosomes that have a problem somewhere in here in the acidic patch, you can see a dramatic decrease in the ability of this wild type protein to tether, the SSX protein to tether to the nucleosome. And this was further shown by incubating and performing competition experiments with a very well-known nucleosome acidic patch binding protein called the LANA protein. And you can see that this is able to outcompete SSX over increasing concentrations. So I showed you that SSX now binds directly to the nucleosome acidic patch. And this was actually an interesting way by which these studies that we had ongoing in synovial sarcoma converged with another student studies in the lab focused on intellectual disability and focused on the subunit SMARCB1. So in parallel with the studies I'm describing to you here, another student in the lab, Alfredo Valencia, was studying this SMARCB1 subunit. And he was particularly interested in this because in a rare but well-characterized intellectual disability syndrome called Coffin-Sears syndrome, he noted that there was a pileup of mutations, particularly at this C-terminal region, this putative coiled coil region of the SMARCB1 BAF47 subunit. There are 9 independent cases, for example, of individuals with intellectual disability that had this deletion of this positively charged lysine, and also any other kinds of mutations in here that are, again, a positively charged residue being swapped out for a different type of residue. And what he found was that this particular region of SMARCB1 was actually important for interfacing with the nucleosome. And to put things in context, before the study, there was no knowledge as to all the different components I told you on BAF complexes. There was no knowledge as to which components actually hit the nucleosome. We know the substrate of a BAF complex is the nucleosome, but we had no idea of all the subunits there, which ones actually touch the nucleosome, with the exception of the ATPase. And so what we found was that this little minimal region, just if you make this region of SMARCB1, this minimal region was sufficient to bind nucleosomes. But if we made any of these disease-associated mutations, these Coffin-Sears associated mutations, you could see here very cleanly that all of these completely inhibited the ability of SMARCB1 to bind the nucleosome. And the net result of all of that was that if you perturb this C-terminal region, there's a decrease in the ability of these complexes to remodel chromatin. So they don't function as well. And that's the disease mechanism for intellectual disability that are driven by these mutations. And we had a story on that that was published last year. Very excitingly, if we now take our new structure that was just published last week, you can see that there are a number of mutations, not only in intellectual disability, but also in cancer that pile up, just single residue mutations. And many of these break this key interface of what we solved as an alpha helix, binding the H2A H2B interface of the nucleosome acidic patch. So this is how the complex grips onto the nucleosome acidic patch. And you can see very frequent mutations in cancer that actually will change this residue from a positive charge to a negative charge or a neutral charge, and break the ability of this helix to properly form and engage with its counterpart here on the nucleosome acidic patch, this very nice negatively charged pocket that's created on both surfaces of the nucleosome. So to try to understand how this might interplay with what I've been telling you about synovial sarcoma, because we knew that the SS18-SSX fusion oncoprotein for the past 5 years, we and others showed that this resulted in the decreased stabilization of SMART-B1. We wanted to understand maybe this would have an impact, this ability of SSX to bind the acidic patch and SMART-B1 to bind the same acidic patch. Would this start to explain why we see the apparent ejection of this complex of this subunit from the complex? So what we did is we incubated nucleosomes with a biotinylated SMART-B1 that just contains that C-terminal region I was telling you about, and we sought to compete that out with increasing concentrations of SSX. And indeed, as you can see from this gel, over increasing concentrations, even at very low concentrations, SMART-B1 can be very quickly outcompeted by SSX. However, the reverse is not true. So if we now incubate nucleosomes with SSX and try to outcompete them dominantly with as much SMART-B1 as possible, you can see here on this Western blot that you're unable to do that. So this maybe starts to explain the mechanism of dominance, why the SS18-SSX fusion protein is dominant in these complexes, and how this may play a role in the destabilization of SMART-B1 on the nucleosome, and maybe ultimate destabilization on the complex, leading to partial degradation. So this is what I'm describing again, as I told you in the earlier work that we had done. You can see here that the fusion protein usurps these complexes and causes for the degradation and displacement of BAF47. These were the original observations in our 2013 study on this, where again, you see it's not absent, but severely decreased levels of this BAF47 protein, both on the complexes itself, as well as in the total protein levels. So what happens when this SSX engages the nucleosome acidic patch and disrupts this SMART-B1 binding interaction with the nucleosomes? The net result is that these complexes can actually still remodel. So they still have an acidic patch binding site at the bottom. They can still remodel. They're remodeling at a little bit of a lower remodeling percentage here, remodeling frequency, but it's enough to still elicit changes in DNA accessibility over these sites that are normally repressed. So you can see here over SS18-SSX target sites, the accessibility of those sites upon gaining the fusion goes up. And you can see that that is also the case here if we look at these new sites that are targeted on chromatin. So all of this is exciting and shows the very strong binding to the nucleosomes, shows this very interesting and cancer-specific property that the SSX 78 amino acid confers for BAF complexes, that they can bind very, very tightly to nucleosomes via the acidic patch. But the acidic patch doesn't explain still, why do these BAF complexes target selectively to repressed regions on chromatin? Why are they going to these PRC2 target sites and PRC1 target sites? We don't understand that. And the acidic patch would be insufficient to explain that because, of course, the nucleosome acidic patch is on every nucleosome throughout chromatin. So what's the basis of the specificity there? So we turn to take genome scale shRNA and CRISPR screening strategies that were looking at proliferative fitness. And here we just subjected the SYL1 synovial sarcoma cell line and compared it to this histologic mimic SW982, which does not have the fusion oncoprotein, and look for differences in dependence on various factors. And among the top dependencies in synovial sarcoma were dependencies on ring 1, ring 1A, ring 1B, PCGF3, PCGF5. These are members of the PRC1 polycomb repressive complex 1 complex that plays a role in repression. And so you can see here now also taking Project Drive, another shRNA driven effort where you're looking at the various synovial sarcoma cell lines shown here. You can see again that some of the top cell lines that are sensitive to depletion of either ring 1B or PCGF3 are actually these synovial sarcoma cell lines. They're at the very top of the curve, and some of these are at the very, very highest mark. So to try to examine this further, we sought to examine whether SH18SSX fusion protein containing complexes, as well as the mark placed by PRC1 repressive complexes, H2A ubiquitination, would overlap on the genome. And you can see here from the ChIP-seq studies that indeed, really the H2A ubiquitination, the enrichment there is very much over the sites of SH18SSX1 over the genome. You can examine this a little bit further by zooming in on a certain locus. So here's the SLT3 locus, for example, and you can see that in the context of the fusion oncoprotein, so when we have not knocked it down in these synoviosarcoma cells, this very nicely overlaps with H2A ubiquitination, the mark that's placed by ring 1A and ring 1B of PRC1 and PRC1 itself, marked here by ring 1B. We then turn to the structure of PRC1. From Song Tan's group, where we were able to then use the structure to inform mutagenesis approaches to break various functions of PRC1. Particularly, we wanted to break the function of the complex in placing the H2A ubiquitination repressive mark, and to examine then what happens if that repressive mark is no longer on chromatin. Are these SH18SSX bound complexes still targeted to the sites? And what you can appreciate from the immunofluorescence shown on the right is that when we made these mutations, for example, compared to the wild type, when we made this mutation in ring 1B, this R91E, or this dual mutation, I53A, D56K, you can see that the localization of SH18SSX is no longer co-localizing with these regions of repression shown and marked by ring 1B. So even when they had ring 1B complex localizing, you can see there's no longer the ability of that complex to localize. So it's of the synoviosarcoma containing SH18SSX containing BAF complex to localize to these sites. So when the histone mark is no longer present, but the complex is still present, it can no longer go to those sites of repression. So this begs the question, of course, what region of that 78 amino acids is actually mediating this clear preference it has for these sites of H2A ubiquitination? So to that we purified now GST, tagged SSX1, so 78 amino acids, and just now incubated the 78 amino acids of SSX1 with either wild type nucleosomes or nucleosomes that have this repressive mark on them. So the H2A lysine 119 ubiquitination. And you can see here a very clear preference when we try to incubate the 78 amino acids with regular nucleosomes or nucleosomes that have this histone mark. You can see very clear preference for the nucleosomes that have this H2A ubiquitination, perhaps starting to explain why there's a preference and overall skewing of these complexes to these repressed sites. And if we examine the sequence a little bit, again, in detail, as I showed you before, we know that this basic region I showed you is important for binding the nucleosome altogether. It actually engages the acidic patch, but we didn't know what this acidic region did. We knew it was important in cells for the targeting of complexes, but we didn't know biochemically what it might be doing. So to try to examine whether this last 7 amino acids or so were actually responsible for this preference to H2A ubiquitinated nucleosomes, we created a variant of this where we mutated all of these to alanines. And what you can see here is if we compare now wild type SSX compared to SSX with the mutated acidic region, you can see that this relieves the preference. The preference for H2A ubiquitinated nucleosomes. So in the wild type, they again prefer, in this experiment, they prefer the H2A ubiquitinated nucleosomes. However, upon removal of that acidic region, you can see they no longer prefer those regions. So something about this acidic region here in the C-terminus is required for the preference to go toward H2A ubiquitinated nucleosomes. So as a model for the story I've shown you, what we see here is that by virtue of this chromosomal translocation, SS18 wild type, which is normally a member of BAF complexes, turns into SS18SSX. This SS18SSX fusion protein then binds into the complexes, particularly by the ATPase module. It disrupts the core, particularly the core that contains the SMART-B1 or BAF47 subunit. And the core disruption is mediated by the fact that this SSX anchor is now binding aberrantly to the acidic patch in place of the normal acidic patch binding element from the core, which is the SMART-B1 C-terminus, as I showed you. And so with that, I'll thank the members of the lab that did the work. This was led by a very talented graduate student, Matt McBride, who is now a postdoc at Princeton, and a postdoc in the lab, Nizar Mashtalir. These two worked very well together to derive this mechanism. They were aided by computationally by Drew Davino in the lab, Martin Filipovsky and Evan Winter, and we're very grateful to our collaborators in Tom Muir's lab, particularly Hai Dao, for the development of this photo cross-linking approach, which was really useful for us in identifying the interaction of the SSX tail with the nucleosome acidic patch, and all of my funding sources. Thank you very much.
Thank you. Thank you so much. Wow. Sorry, I couldn't get unmuted on there. We have a couple questions that have already come in. There'll probably be some more as we answer these. Thank you for the great talk. So the first question we have is, for the combinatorial assembly, are there requisite components that are always present in the final complex? If there are these, I just lost the end of it. Are these more conserved or less subject to mutations?
That's a great question. So there are complex components that are present in every one of the assemblies, and they must be there for the entire complex to form. So in our 2018 cell paper that we had worked out the order of assembly and the modular organization, one of the interesting findings to emerge from that was that there's actually a homo or heterodimer that forms, and that's the platform for the entire complex to be formed and to be put together. And so what we can see there is that there are SMARC-C1 and SMARC-D1 subunits that are present there in that early core of the complex, and yet you need that core to be able to form the complex. So yes, you actually are absolutely correct that mutations in that core are far less frequent because they would be all too deleterious. In fact, what we found is that there are more mutations as you go through and progress through assembly. The mutations tend to skew at later parts of the assembly pathway, because if they occurred too early, all the complexes would be wiped out. It's all too deleterious for the cell. It's a great question.
Thank you. Next question is, what is the subcellular localization of either SS18 or SSX alone?
So SS18 itself is a member of the BAF complex or switch in a family of chromatin remodelers. So all those are always nuclear. So SS18 is always nuclear. SSX, when it's fused to SS18 by virtue of the fusion oncoprotein, is also nuclear. It's not present in the cytosol. All these are nuclear. SSX itself is actually a testes-specific protein. So it's normally never expressed in any of our adult tissues, with the exception of testes and with low levels in the thyroid. So this is a very aberrant. This really creates for the aberrant fusion protein. This is a protein that's not supposed to be expressed in any of our adult tissues, and so its localization is also nuclear.
Okay. Next question: acidic patch binding is attenuated, not eliminated, by mutating away the acidic residues. Can Dr. Kadoch comment further on this, and would we expect any retention of functionality?
Yeah, it's a great question. So the ability of SSX to bind the nucleosome is completely dictated by the acidic patch. It does engage the acidic patch. So if we do break for example, one of the arginines there in that region, you do see a complete loss of the ability to bind to the nucleosomes. If you make these single residue mutations, you can see that they fall off nucleosomes altogether. And that's because you're disrupting the positive charge that's required to engage that negative charge. So in most of our experiments, I believe this was a near complete removal of the binding interaction between that and the acidic patch. You can imagine certain point mutations may partially stabilize or mostly destabilize, but this just depends on which mutation we make within the acidic region.
Thank you. Next question is, how accessible are PRC1 sites still bearing H2 AUB119 bound by SS18 SSX BAF compared to non-PRC1 SS18 SSX BAF sites?
How accessible are they? So they are the H2A mark sites over which these BAF complexes localize are more accessible than the H2A just by itself, the H2A ubiquitinated sites that are alone without BAF complexes. It's a little hard to discern that because, again, these complexes are dynamic. So you see a lot of on-off rates. But if you look at the pileup of complexes containing the fusion over the H2A ubiquitinated sites, the majority of those sites where the fusion is present are more open than those that are not. It's a good way of doing the analysis. I like it.
Next question asks if you're able to comment on the upcoming structural study and what resolution was the structure.
On this structural study, so our structural study is actually at lower resolution, and the reason for that was because we took a combined approach and actually purified these complexes out of cells endogenously. So this is the first structure of an endogenous human BAF complex. This was taking purified complexes from cells, and so keeping and trying to keep the complex in its native form, with and without binding to a nucleosome. So the resolution was more poor, but we were able to combine that with homology modeling for yeast structures and also individual components to achieve the resolution that we achieved.
So the next question is, why is the SSX fusion so specific to synovial sarcoma? And then part 2, does the H2A 119K UB dependence suggest that PRC1 landscapes in the cell of origin mediate the context specificity? And then also, you want to do those two first?
Sure, I'll take those two. So that's a great question, and you're right on. We absolutely think that the PRC1 landscape will play a role in making a cell permissive or not permissive of this fusion oncoprotein. Of course, a huge question in the sarcoma fields in general are just cell of origin. It's a majorly important area, one that our lab doesn't focus on too much, but a lot of labs throughout the world are focused very heavily on this. You know, it clearly suggests that there must be a very specific cell of origin that is uniquely permissive of the fusion event, because it's quite possible that this fusion event happens in other cells, but it's not selected for. And that can actually be seen in the lab, because if we take the fusion oncoprotein and put it in certain types of fibroblasts, the fibroblasts die. In fact, you can use senescence assays to actually measure the impact of the fusion protein. But if you take just a different kind of set of lines, or you can increase the proliferation rate by 4 to 8x. So it just depends on the context. And so the actual feature that is most dominant in determining whether or not the cell is permissive of the fusion protein and it will lead to proliferation or not remains to be determined. And it's a great question.
Okay, and then the third part of that question is, would this mean EZH2 inhibitors are ineffective in synovial sarcoma?
Yes, it does. That's a great question as well. And actually, they were tried. Unfortunately, they were tried for synovial sarcoma. What our mechanism argues is that, in contrast to malignant rhabdoid tumor or ATRT, where these mutations affect BAF complexes and they're no longer able to function. For example, you miss the entire SMARP1 subunit, these complexes are essentially shot and they're unable to function. That's an example where EZH2 inhibitors may have utility. And in fact, that's exactly where the EZH2 inhibitors have shown some utility. However, in this case, this is a gain of function mechanism. If anything, the last thing you want to do is further inhibit polycomb, because what you're doing is this SSX is dragging complexes to displace the residual polycomb that's there. You're just removing the remaining repression over those sites. And so that's one of the reasons why we think these inhibitors have been unsuccessful in this gain of function setting of synovial sarcoma.
Well, that was our last question. So I just want to thank you again for all that you do and giving us this talk and giving us your time today.
And most importantly, I want to thank you, Jay, and the entire ALSF for supporting our research for many, many years now. In fact, this was my very first award that I ever was able to receive upon starting my lab. In fact, it was actually before I even moved across the country to start the lab. So I'm very grateful for the support of the ALSF and most recently the support through the A Award. This has meant a great deal to our research program and continues to propel forward very exciting discoveries in this area, not only for synovial sarcoma, but for many other fusion oncoproteins. So thank you.
Thank you. And then we can say we knew you when.
Thank you so much for the opportunity. Have a great day, everybody.
I appreciate it and take care.
Bye-bye.